Hey everybody. Hey everybody, welcome to chapter 8. So we're going to continue on in our lecture. We started in chapter 7 talking about bacterial genetics, and now we're going to talk about how mutations can either improve or can potentially kill off bacteria. So bacteria have to be able to adapt to environmental conditions, and one mechanism of adaptation is via mutation. A mutation is a change on the genetic level. And so I found this little figure on Google Images. I apologize, I didn't get the actual website. But here is an example of mutation producing disease or bacteria that are resistant to a certain drug. So here's some cell, parent's progeny, produces lots and lots of offspring. Some of these offsprings have a mutation. So bacteria multiply, there's billions of cells in the body, and a doctor prescribes some drug, some antibiotic. Patient takes the antibiotic and it kills the cells that are not resistant to it, but those cells that are resistant survive and then they multiply. And so now we have these drug-resistant cells that are thriving. So that is one mechanism of how mutations can be potentially detrimental and, well, beneficial, depending on if you're the bacterial cell or if you're the organism that is infected. Mutations can be transferred from parent to offspring. So you have one parent has some mutation that occurs spontaneously, and they pass that mutation on to their offspring. Another mechanism of mutation transfer would be horizontal. So the first was vertical transfer from parent to offspring. The second is horizontal, where you have two cells in the same generation that are sharing information. And so in this form, we have a cell that contains some plasmid. We have a cell that doesn't contain a plasmid. So they come together and this cell shares its plasmid, and now both cells have a plasmid. So what's really crazy is that you don't have to be the same species to share plasmids. You can be Clostridium perfringens and Staphylococcus aureus could share information. Total different species. They live in different environments, but they could share genetic information through horizontal gene transfer. So a mutation changes the phenotype of an organism oftentimes. Mutations, so a mutation on the gene changes the genes, and that can change the physical characteristics. So remember, genotype is the genetic information of the organism. What do the genes say? Phenotype is the physical characteristics. So what is actually being expressed? Deletion of a gene for tryptophan, example, would produce a mutant that can only grow if tryptophan is supplied. And in this case, the mutant would be called an auxotroph. Auxotrophs are organisms that need certain types of growth factors, and they can't grow without those growth factors. The prototroph is the original or the parent that did not require the growth factor. And so we often produce auxotrophic cells so that we can test different substances on those cells, which we'll talk about in a little while. So you can have spontaneous mutations, or you can have mutations that are produced. So a spontaneous mutation is a mutation that occurs through normal processes. So, I mean, mutations happen. DNA replication isn't perfect. It's 99% perfect, but not perfect. And so, though the mutation rate is rare, it still can occur. And spontaneous mutations are low frequency mutations, but when they do occur, they can potentially cause beneficial changes to an organism, and oftentimes they can cause organisms to have detrimental qualities that make them less likely to survive in an environment. So when you have a large population of cells within a colony, almost always you're going to have some cells that are not identical to all the others. The majority will be one type of cell, and then you'll have a few that are called mutants. In spontaneous mutations, the environment isn't causing the mutation, but sometimes the environment selects those organisms that are mutated, which means they are more likely to survive in the environment. So organisms that have a certain resistance factor typically end up becoming more common than those that don't have that resistance factor. So there are 3 types of spontaneous mutations that occur that are called point mutations. So point mutations are mutations where one nucleotide in the entire strand of DNA changes. And when that one nucleotide changes, then it can cause different problems, potential problems. So with a point mutation, also known as a base substitution, you can have a reading frame, so that you have that reading frame where you have the 3 nucleotides that are supposed to produce some certain amino acid, instead produce a nonsense code. And that's also known as a knockout mutation. You can have a mutation where the nucleotide change doesn't actually change the amino acid that is coded for, due to the degeneracy of the code or the number of codes that code for the same amino acid. That's known as a silent mutation. And then there are mutations where that one nucleotide change changes the amino acid that is produced, and that's called a missense mutation. So here is a picture of DNA. Right here, you see within the DNA, you have a TGT and an ACA, okay? So we're looking at TGT is the code, and HCA is what we're using to produce an amino acid, okay, or to produce the messenger RNA. So here we have the wild type, TGT. ACA is the template. ACA produces a UGU, which is going to code for cysteine, which is what that TGT codes for, okay? So next to this, we have TGC. So we have one nucleotide change in that 3rd position. So now the template says ACG, so now you produce a UGC, which is again identical to that coding strand. We still produce cysteine, so that's a silent mutation. In that 3rd position, now we instead have a TGG, so still one nucleotide change. Template now is ACC, so we produce a UGG. UGG actually codes for, and so TGG codes for tryptophan, which is a different amino acid than cysteine. Therefore, this is a missense mutation. We miscommunicate. And we now have a different amino acid put in to that polypeptide, and that changes the functionality of the entire protein. And then the last one, you have a TGA instead of TGT. So now your template says ACT, and your messenger RNA is UGA. UGA actually codes for a stop site. So translation terminator sequence. And so that's going to be that nonsense or knockout mutation that stops translation from occurring. I'm going to stop here and we'll talk about deletion and addition mutations in our next video. Bye.